Our next speaker is Dr. Aaron Mansfield from the Mayo Clinic in Rochester, Minnesota. Dr. Mansfield received his MD from the University of California, Irvine School of Medicine. He completed his residency and fellowship at the Mayo Clinic College of Medicine and is now a clinical investigator in the Internal Medicine Hematology Medical Oncology Division at Mayo and an associate professor of oncology. Dr. Mansfield is a translational scientist with a focus on early phase clinical trials, especially for lung cancer and mesothelioma. Dr. Mansfield's research is focused on understanding how tumors avoid destruction by the immune system, and he hopes to introduce novel treatments and tools to treat lung cancer, mesothelioma, and their associated complications. And he'll be sharing with us his work on neo-antigen potential of chromosomal rearrangements and mesothelioma.
Well, thanks for the opportunity to present today. I am excited to share this with you. I've enjoyed your other presentations, and thank you for bringing me out of my field of oncology. But it's nice to see how immunologic principles hold regardless of the system being studied. So I hope to discuss with you the current treatment landscape for mesothelioma with immunotherapies. That is actually changing for the better. I'll show you some of those data. But then there's a clinical conundrum in mesothelioma with how tumor mutation burden is really low, but this is an immunotherapy responsive tumor. I'll get into that in more detail. And then I'll cover what I think are alternatives to non-synonymous base substitutions as sources of neoantigens, which for at least those in oncology, I hope you find relevant to your work. That being said, since many of you may not see mesothelioma, on the far left here, this is unfortunately from a patient who's passed away, but that's looking at this patient's lung. In white around the lung, you see the actual tumor, the mesothelioma. You can see it expanding into the fissures. This restricts their breathing. Most of the time, this doesn't metastasize, but you'll see it completely encase the lung and restrict their breathing. And we know this is primarily due to asbestos. There are other causes, one being radiation, and there's also a genomic predisposition syndrome in patients with germline BAP1 mutations. But in the middle, you have an electron micrography of asbestos minerals, and it was a physician in South Africa in the late 60s who really made the association between asbestos exposure and the development of mesothelioma. The incidence of mesothelioma was directly correlated to the tons of asbestos that were mined. There's a many-decade lag between exposure and development, and with most of the miners being male, but most cases are in males. In the US, at least, most of my patients were in the Navy and were exposed to asbestos that way. In the labs I visited in the UK and in Australia, more of them were carpenters. Just a little more background, then I'll get into more of the work we've done. But I've spent time in Perth, Australia, and in Western Australia, they have one of the largest asbestos mines in the world, which is now abandoned, called Wittenoom. So on the left, that top strand of this rock, that's blue asbestos. This mine, Wittenoom, is known for having this blue asbestos product. The picture on the right is not doctored. That's what that asbestos looks like. When it's ground, it sort of looks like sand. These poor kids died in their 30s of mesothelioma. Father was a miner. Their fathers were miners, and the kids would play in asbestos boxes the way we would in sand boxes. So they died in their 30s, having been exposed when they're really young in life. So, so moving on, I'll show you data that immunotherapy is superior to chemotherapy for mesothelioma. I'll show you why PD-L1 and tumor mutation burden are imperfect for figuring out who's going to benefit from this disease. And then I'll show you what we've done to try to get us a step closer to figuring out who does benefit from PD-1, PD-L1, or CTLA-4 inhibitors. So we got the impression that immunotherapy with the immune checkpoint inhibitors would be beneficial in this disease. We published this 6 years ago, but we looked at a large series of patients with mesothelioma and looked at their tumors and assessed for PD-L1. And we showed that PD-L1 was more commonly expressed in a variant of mesothelioma, and it was a negative prognostic marker. And this is a finding that's been shown by many others with very similar frequencies and survival outcomes since. These curves go to show just how abysmal of a diagnosis this is. The median is around a year to 14, 16 months based on what series you're looking at. But we got some signs of hope as these immune checkpoint inhibitors were developed. These are data with the PD-1 inhibitor pembrolizumab. This was a non-randomized, just single-arm phase 2 study. It treated patients after they had already received chemotherapy. And when we counsel patients in clinic, we tell them the survival in clinical trials, the average is about a year with chemotherapy. So these patients had already received chemo and they were getting second or third lines of therapy, and their median survival was 18 months. So after chemo, they lived another 18 months on average. So what we had never seen a benefit of this degree in the disease, and this just spoke to the potential for immune checkpoint inhibitors to improve outcomes. So similar work was done in France. This is looking at nivolumab, the PD-1 inhibitor, or nivolumab with the CTLA-4 inhibitor, ipilimumab. This was a randomized study, but the goal was not to compare the two treatments. It was just to assess the efficacy of both of them. And nivolumab plus ipilimumab did seem to be a little bit better, but again, it wasn't powered to look at a difference between the two. And just like the data I had just shown you, we're seeing after frontline chemotherapy, we're seeing survival outcomes, medians extend beyond a year in the second or third line setting, which again, just is something that is essentially unheard of in this disease, and very promising. But when you try to ask, how do you predict who's going to benefit and try to select which patients to get these therapies, it gets tough. Some of the side effects with these therapies can be fatal with pneumonitis. A patient of mine just had a colectomy for colitis that would not resolve despite every immunosuppressive treatment we had to give her. But here's looking at the French data where they took PD-L1 expression, they used a 1% cutoff or a 25% cutoff, and PD-L1 expression at either cutoff did enrich for responses to nivolumab or ipi with nivo. But the problem is patients who had no detectable PD-L1 expression based on that 1% or 25% cutoff still responded to treatment. So even patients without detectable treatment or below your threshold for your cutoff, they can still benefit from immunotherapy. And we try to look into this further, and my surgeons like to operate, so I've got a lot of tissue to work with. And here we took cases that had multiple sites of disease that were resected or had late recurrences after surgery. So the 3 columns on the left are looking at the H&Es and PD-L1 expression of the primary tumors, and the 3 columns on the right are looking at the H&Es and PD-L1 expression at the paired distant lesions or the recurrent disease. And each row is a case. So here's a patient with a sarcomatoid mesothelioma. PD-L1 expression was present in the primary site and the distant site. But then here's a patient with epithelioid mesothelioma. The tumor cells were highly positive for PD-L1, but the distant site, it was only the immune cells that had PD-L1 expression. And here's a separate patient where the immune infiltrates were highly positive for PD-L1, but the distant site, only the tumor cells were positive. So in the end, the kappa for agreement in our hands with mesothelioma was 0.4, which I think is really inadequate to use as a biomarker for what that's worth. So this just goes to show if you stick a needle in one spot instead of another, you might miss expression, and that's what makes using PD-L1 difficult. I think in this disease, we have similar data published in non-small cell lung cancer showing the same thing, and it just challenges the use of a biomarker that is dynamically and adaptively expressed. So that being said, we just presented data. I know we had some investigators from the Netherlands Cancer Institute earlier, and my colleague there, Paul Bass, presented this just a month ago at our now virtual World Congress on Lung Cancer meeting that would have been in Singapore it weren't for COVID. But we ran a phase 3 study with Bristol Myers Squibb, and we took over 600 patients. They were randomized 1-to-1 to receive the PD-1 inhibitor nivolumab with the CTLA-4 inhibitor ipilimumab, or a platinum doublet chemotherapy, which has been our standard for almost 20 years. And I'm not going to go over all the data that take the whole meeting, but the short of the story is we showed that the immunotherapy combination is superior to chemotherapy. For all comers, we improved the median by 4 months in terms of overall survival, hazard ratio of 0.74, p-value of 0.002. So this was not just statistically significant, but I think clinically meaningful too. And hopefully in another year or two, we'll see what sort of tell of a curve we have as more as the data mature with the follow-up outcomes. But we expect this to receive regulatory approval based on these data. This is under review for publication at New England right now. So hopefully these are in manuscript form by the end of the year for everyone to see. So this is moving forward. But we've done work looking at patients who had surgical resections, but I'll come back to these data later in the talk. But the conundrum for mesothelioma is, even though it's a cancer related to carcinogens in over 90% of the patients, it has one of the lowest tumor mutation burdens for an adult malignancy, especially one that's defined by a carcinogen exposure. So this is a different plot than what you typically see in that Nature paper with Alexandrov as a author there. But when you look, you've got on the far right squamous cell of the lung, then small cell lung cancer, bladder cancer, adenocarcinoma of the lung, tumors that we often see benefit with immunotherapy. They have about an order of magnitude higher tumor mutation burden than mesothelioma. So the data on top come from Genentech and Rafael Bueno at Harvard. They did this study jointly in mesothelioma and aligned this with some other data from Genentech and some publicly available data to come up with this plot. And then TCGA just 2 years ago published their own genomic data set on mesothelioma, and they said that the tumor mutation burden was less than 2 per megabase in all samples except one that had an MSH2 driven phenotype. So there's multiple lines of evidence suggesting this has very low tumor mutation burden. And where this goes against our understanding of benefit to immunotherapies, the FDA just approved pembrolizumab, the PD-1 inhibitor, for patients with 10 or more mutations per megabase. So why is it we're seeing all this evidence that immunotherapy is beneficial in mesothelioma, and yet we're being told there's so few mutations? It just wasn't adding up to us as these data were emerging. So we spent the last 3 years really looking into this. And then the other line of evidence to support that this might actually be a wrong assumption is when you go back to the 80s and 90s and you look at karyotypes. Mesothelioma does not have, it's hard to find a normal chromosome in this disease. There are recurrent chromosomal gains and losses that have repeatedly been demonstrated with karyotyping that we don't do as commonly anymore as our genomics have improved. But you have to ask, what are your genomics capable of detecting? And can it really detect these structural variants? And I would argue that the most common platforms do not. So sorry, Zoom's giving me a notification. While we were doing our work, Michela Carboni and a group in Japan led by Dr. Yoshikawa, they were investigating this genomic syndrome I mentioned earlier, BAP1, where these BRCA-associated protein 1 is often lost in families with multiple family members with mesothelioma or uveal melanoma or renal cell carcinomas. And there's a discrepancy where genomics, we're not finding BAP1 mutations, but by immunohistochemistry, we could see that the majority of tumors lose BAP1 expression. So they designed a high-density array CGH and showed that this part of the chromosome where BAP1 is on a 3p21 is often involved in areas where there's alternating gains and losses, and they suggested that this was a chromothriptic involvement, which your standard genomics may miss. I'll get into what chromothripsis is in a few more slides, just for those who aren't familiar with that. But this paper gave us the suggestion that we were on the right track with what we were doing at the time. Our lab and our bioinformatics team had really optimized an approach called mate pair sequencing, which is Illumina is going to stop making the primers for, but that looks at longer DNA fragments, anywhere from like 2,000 to 5,000 kilobase or base pairs, which gives you much more extensive coverage. And I'll show in one of the next two slides how that improves your ability to detect a rearrangement. We then, once we found rearrangements, used RNA-seq to validate that they were expressed. We then used PCR and Sanger sequencing to validate specific rearrangements. We then used our genomic data to HLA type our patients, and then we looked at these rearrangements in the context of immunity and ran them through IEDP and NetMHC to see if some of these would be predicted to be presented by these patient-specific MHC proteins. ProImmune helped us with the REVEAL assay, looking to see if some of these peptides or neo-antigens actually bound to the MHC proteins. And then we did ELISpot to confirm that there were actually reactive T cells to these neo-antigens. I'm not going to show the Immunoseq data, but we did profile the T cell clonality. Just for sake of time, I'm going to skip our pipeline. I'm happy to share this with anyone. It's also in our publication. When you look at standard sequencing and you look at your bridge coverage explains sort of the length of the genome you're covering. And here, if you look at a standard sequencing approach with, say, 500 base pair fragments, and you look at 15 paired end reads, you may only have bridge coverage of 5 times to detect a rearrangement. Whereas when we're looking, we're using DNA that's 2,000 to 5,000 base pairs, your ability to cover a rearrangement goes up an order of magnitude. And in our approach, using primarily frozen tissue, but now we've developed an FFPE approach, and that manuscript is under review. So hopefully, again, it'll be shared in the near future. We're getting anywhere from 40 to 100 times bridge coverage to detect rearrangements, and we've almost always been able to validate them both at the DNA and RNA levels. So that being said, I know Circo's diagrams are in favor. My bioinformatics team prefers genome plots, where you look at the chromosomes 1 through 12 on the left, and you come over to the right, and you have 13 through 22. This just gives you a little better resolution to look at the rearrangements. Here's a sample case where we have a rearrangement between 2q and 8q, and then we have a junction plot where you can see exactly where these junctions are between these two chromosomes. This one's bidirectional, so this was a balanced translocation. We had 76 times coverage to detect this. So this is just an example of the type of readout we get. Some tumors just have a single rearrangement, others don't have any. It really depends on the tumor type you're looking at. But in mesotheliomas, I'll show you in the next few slides, we see many. And I'll use a metaphor, just an encyclopedia as I'm explaining some of these events. So if you think of our set of chromosomes, what we have, just imagine that we had a corresponding encyclopedic encyclopedia set of books, but 2 copies of each volume. So here's one case of mesothelioma, lots of rearrangements, and then you look at chromosome 21. This thing is completely rearranged with alternating gains and losses. That's something that's called chromothrypsis. This is very similar to what our colleagues called out in BAP1 in this disease. And we liken this to taking a volume or a chromosome, if you're using that encyclopedia metaphor, removing many of the chapters, rearranging them, and throwing a few of them out. The other thing we often also see in mesothelioma is chromoplexy, where multiple chromosomes are involved with rearrangement. So here, I've got a chromothryptic event on 7 that's involved with chromosome 11 and 16. So we liken this to taking multiple chapters from different books and assembling them into one of those books. We have prior publications showing that this can drive known oncogenes. I published on this being a driver of ALK rearrangements in sarcomas, work I'm not a part of, but my bioinformatics team is, looking at HER2 and breast cancer. They're very frequently involved with rearrangements that hit HER2. Mesothelioma, we were interested in this though for its neo-antigenic potential. And I'm going to skip this slide because I know I'm short on time, but the black circles that are filled with white just show you the number of counts we were seeing per specimen. The black ones show you how many of these involved genes, and then the ones in green show you how many of those rearrangements we had RNA evidence for them being actually transcribed. So here is one of the rearrangements. This was from an intrachromosomal chromothryptic rearrangement involving CADPS2 and an intron. So this was from our mate pair sequencing that we had 44 times coverage to suggest this was a real event. We developed primers that span the junction to validate it was there at the DNA level. We had done RNA-seq, but we also developed RNA primers, looked that it was expressed at the RNA level too, and then at Sanger sequencing, we confirmed exactly where this junction occurs between this intrachromosomal rearrangement. Like I said, this is typically non-coding DNA that we're seeing end up downstream of a gene that is being expressed. So this gave us the indication that some mesotheliomas might be expressing some quote unquote junk DNA or DNA that to our knowledge typically is not transcribed. And we're wondering if that could be neo-antigenic. So we here, that same rearrangement I showed you, we took the HLA profile of that patient. Here it's just repeating at times, but then here was that sequence of peptides that were coded by the typically non-coding DNA, and we looked at the amino acid sequences, and we used, again, NetMHC and IADP to try to figure out which of these may get presented by these MHC proteins and what the immunogenicity was. We're still working on some visualization approaches to this, but the larger the figure, be it the circle or the triangles, the more RNA transcripts we have to suggest that these are actually being transcribed on RNA-seq. And then the colors relate to what HLA molecule or the HLA that could potentially bind those peptides. So again, this is work in progress, but we selected peptides that seem to be presented by multiple MHC proteins, not just a single one. So they actually had a chance of being presented to the immune system. I know the reverse approach was being taken from some of the data presented earlier, but here we're looking at generating immune responses. So here's another way we graph this, looking at immunogenicity, incorporating the proteasome and TAP processing scores in light of the HLA type that the patients had. Again, ProImmune really helped with looking at the binding. We took peptides that, to our knowledge, are not expressed in humans, and but we worked on our 3 favorite ones from this patient. And in comparison to the positive control peptide, I mean, they all seem to bind as well or almost as well to the HLA molecules of interest from this patient. Looking at the LE spots, in this patient, we also found evidence that a number of our predicted peptides had T cells that could recognize and react to them, some much more avidly than others. We used a very similar LE spot approach to what Dr. Knowlton described in the last presentation, with the similar controls for what that's worth. But we have someone in-house who's helped us with that. So our ongoing, that work has been published, but that's led to some funding where, in collaboration with Paul Bass, who presented the ipi-nivo data for BMS, we have the pre-treatment and on-treatment biopsies from about 70 patients who received nivolumab with or without ipilimumab on 2 different phase 2 studies. So we're using those biopsies to identify the rearrangements, look at their expression, and see if those predict a response to immunotherapy. And we're also using a similar genomics approach, but more so to study the dynamics of gene expression and any gene rearrangements following the selective pressure of immunotherapy. We also have their peripheral mononuclear cells. Those were not cryopreserved for ELI spots, but we're using, we're doing the T cell receptor sequencing on those to look at how clonality changes before and after treatment, and to see how the dynamics of those changes, or the heterogeneity indices prior to treatment, may affect response and survival outcomes. One other thing that we're doing, which just received an NIH R21 award, those chromosomal rearrangements, we can actually find them in blood the same way you can find an ALK transcript in blood, or RET or ROS1 on commercial panels to select treatments for patients with non-small cell lung cancer. We don't find that the rearrangements involve oncogenes in mesothelioma, but we can find these as cell-free DNA, and we've proposed that these might be used to improve disease monitoring. This disease is very difficult to image and assess response with. So we're prospectively collecting tissue and blood to do this. So hopefully I'll have more to say about this approach in the near future. So I know I'm right at my time, but I hope I've convinced you that there are frequent chromosomal rearrangements in mesothelioma. In fact, I think there are more rearrangements than there are single nucleotide substitutions. Most of these rearrangements are often in very complex patterns of chromothrypsis or chromoplexy. We see that these can be expressed, and we think some of these may in fact have some potential as being neo-antigens, and hopefully by the end of the year or early next year, I can say whether these predict outcomes in mesothelioma. So with that, I'd be more than happy to take any questions, and I thank you for your time, and again for the opportunity to meet many of you virtually and hear your work and present today. So thank you.
Thank you very much, Dr. Mansfield. Thank you. I just wanted to sort of just probe on the Hla binding assays that you did. So with those particular epitopes, had you have a chance to then monitor those immune responses using multimers as opposed to the ELISpot?
I would love to do that prospectively with, but the problem was the way the PBMCs were collected for the prior work I showed you, and then also for the way the specimens were collected by my collaborators to see if these predict responses. The PBMCs just simply were not cryopreserved to do that type of work. Now for the R21 funding we just got to look at cell-free DNA, our collection process does cryopreserve those PBMCs for the internal cases we have. So we're going to be able to do that moving forward. And so I'd be very excited to work with you on the monitoring the responses with time. With what's in hand, I just don't have the specimens.
Yeah, that's always the critical thing, isn't it?
Yeah.
Thank you so much. I mean, that's been a real insight, certainly for myself, in regards to mesothelioma and you know, the severe effects, particularly with regards to chromosomal rearrangements. That's quite extraordinary. So no, thank you for that.
Thank you again, everyone.